than I'm growing in my lab at UCI and Scott's growing in his, and Steve's growing in his. It's a different product, and it has to be scaled. It can't just be that you make it QA/QC. You have to make it in precisely the same manner that you're going to make it for when you address humans. So you have to then do a market analysis and anticipate that you're going to treat 2,400 patients a year and build a production facility that can handle that, just so that you can go do all of your preclinical animal studies. That's quite a barrier. So here's batch release criteria for oligodendrocytes. It's not small. There's a batch release criteria example from human motor neuron progenitors and what we say we're going to do, and then the results that we got. I'm really pleased with this. Look at that. That's really, really high. And then you have to, of course, do karyotyping. And then you must also realize that you've got to do exactly the same process for your media. It's not enough that you've got to, I'm sorry to mean to be throwing out the daunting preclinical talk, but this is, these are true challenges. You can't forget about the media. Media production, again, is the focus of individual companies. It's very unique skill sets to design and implement. They have to be fully defined, free of rodent reagents. Well, there isn't a single product on the market. That's a bit of a problem. So California Stem Cell, that took the motor neuron program, just put these two out. They had to develop an entire product, 2 different products, to grow up the stem cells and then to transplant. That minimal components, fully defined, etc. And they're going to start selling this stuff. This isn't an advertisement for them, but it's the fact that this therapeutic company had to get into media in order to ensure its survival after it went to the FDA. And the FDA said, no, you need GTP, GMP, media production. All of these things apply. The devil here, after you do develop these things, you have to go repeat all of your preclinical work with that media again. It's not only your cell product, it's that simple little carrier. It's very, very daunting. The clinical synopsis is something that you should, believe it or not, do first when you're looking towards the clinic. It's not the thing that you do last after your preclinical gets you all excited, and you actually do it before anything. And it's the result of multiple focus group meetings. I don't know how many I did in the Geron program, lost count. In the motor neuron program, we've done 14 so far. And these are groups of 10 to 15 experts from all over the world that address one thing or another. How you're going to get the cells in, risk to patient, outcome analysis, site specifications. The list goes on and on and on. There's about there's about 14 things you need to address, and each one of them should have several focus group meetings. They should precede the experimental design, and they should reflect the preclinical studies. So, for instance, if Geron goes to the FDA and says, Kierstead Laboratory has shown this, and we've repeated it, we can make animals walk again if we transplant a thoracic spinal cord, they say, okay, fine, you can only work on thoracic patients. So, should have thought of that earlier. We should have done some cervical studies a lot earlier, shouldn't we have? That's just true. We just didn't. But the FDA prohibited us from working on cervicals. And you've got to select the appropriate patients. One of the points that Naomi put up there, must select appropriate patients from a diverse range of injuries. So we don't have the luxury as we do in preclinical work. And in something like spinal cord injury, drawing up a clinical synopsis, one of the great bears is patient recruitment. You want the thing to work. So, okay, we can't do cervical. Oh, right, we can't do laceration. And so there we are. We've got that population yet left. And then how many of that type are there per year? How many sites are we going to have? Now, how much variability is introduced into the surgical teams working on there? So these are things that are not trivial. They can be overcome, but drawing up a clinical synopsis reduces your market down so much that it may prohibit you from running a clinical trial at all. You should figure that out before you design your preclinical work. The outcome measures, of course, with particular spinal cord injury are particularly daunting. It's only the truth that the cure is not around the corner, but incremental advancements are. I'm very positive that we're going to have some success in spinal cord injury fairly soon. I'm an optimist, for those of you who know me, you know that. But I'm very convinced that we'll have treatment soon. But it is the truth that if they work, they're going to be incremental advances. That means that we require graded outcome measures of fine function. And if we do not have that, we could miss something that is tremendous. If we could do nothing more than restore that, but we just fail to pick it up in Asia-FIM, then we've missed a great opportunity. Slight bladder bowel sexual function return, tremendous benefit, but that could fall right off the charts if our outcome measures are not graded and sensitive enough. And rehabilitation regimes right now are in their infancy for spinal cord injury, and no transplantation technology will work without rehabilitation. None. I really believe that. You must stimulate the system in order to have a transplant regime work. And that begs the final question. There's got to be a continuity of care from the surgery suite to the rehab to the outcome. And if there's not a continuity of care, if there's not a medical doctor that's actually in charge of assigning the rehab and the continuity across these things right through to outcome, it's just not going to work. There's just no way it's going to work. We need that continuity, a concierge type program to spinal cord injury. And I know from one group I've worked with that there is apparently, in their view, no center in the United States that has that continuity yet, according to their criteria. I think it's coming, but I don't think it's impossible to do by any means. But until that continuity of care exists, the likelihood of getting outcomes is zero. And then lastly, financing the program. And this is a major challenge to the clinical translation of spinal cord injuries. The spinal cord injury market is extremely small. At 100% market penetration, which has never been seen for any drug, it's a $300 million a year market, so I'm told. And that's too small for big pharma. It's fine for mid-sized companies. And the risk is very, very high because of difficulties with patient recruitment to fit your very tight clinical synopsis and outcome measures where we need very, very fine graded measures of outcome. The barrier to the IND is very, very high given the long life of spinal cord injured patients. So with the California stem cell motor neuron approach, they decided to go after spinal muscular atrophy first because these babies die so quickly that the barrier to entry is lower. And but in a spinal cord injured population, we have to realize that they don't value your, wow, I'm probably going to get myself in trouble saying this, but the duration of your life is prohibiting them from, it's raising the bar, and your pain and bodily dysfunctions don't lower that bar. So the barrier is actually very, very high given the long lifespan of the individuals. And here's just a tremendous thing in financing that I mentioned twice before with regards to the cell preparation and the media, that your final formulation, you have to repeat it all again. Now try to write your second NIH grant and get it funded again. You won't. And, well, no, somebody will. But you've already done it. You've done your inventing. It's already out there. You've got your paper. To get someone to give you the money to do that again is very, very tough. So where do you turn? You've got to turn to industry, of course. And then that opens up the commercial viability side of it. So with these two companies that I've worked with, Ability Biomedical for something I told you about earlier, I didn't tell you about, I'm sorry, it was the first treatment I developed for acute immediate spinal cord injury that altered the immune response. It's now in clinical testing. 120 humans have been treated with this stuff. And I'm really happy, but I developed it for spinal cord injury with my partner, collaborator, Tom Lane, who was working on MS. So together, we did this, but we tried to get companies to be involved, and they said no. After we developed our final formulation and went back and had to repeat everything again, they said no, sorry, market's too small on those things. So what we had to do is go back and show that it also worked in rheumatoid arthritis, which is a $9 billion a year market. And then also ulcerative colitis, which is a $14 billion a year market. And then they said, okay, we'll take it. And so we're doing the same thing with the motor neuron approach in opening up spinal muscular atrophy, ALS, and spinal cord injury. And I don't mean to sound like a callous capitalist, but it's just the facts. That's just how it works. It's too small, so you've got to have a mind to working in other animal models in order to increase the value of the program such that you garner investment. And I want to end here. I added one that is a little more squishy.